The learning objectives of this lecture is to discuss the transmission pattern of single gene disorder, discuss the important features of autosomal dominant, autosomal recessive, and X-linked disorders, list their examples, and also to discuss the exercises to understand the transmission of Mendelian disorders. So, as you all know, that our human body has multiple cells, and this is a human cell which have different organelles and they have their own function. And the nucleus has chromosome, and this chromosome is the basic unit of life, which is made up of DNA. In turn, DNA is a double helix structure with nucleotides as their building blocks: adenine, guanine, cytosine, and thymine. You have studied in your biochemistry. And the segment of DNA is a gene. So this is a YouTube link. If you want to go through that, you can have a much detailed view about it. So before we proceed towards the autosomal dominant and the recessive disorders and to understand the Mendelian inheritance, I would like to discuss some important terminologies regarding these inherited pattern. That is, let's start with that. Genetics. You know that genetics is a field of biology that studies how characteristics are transferred from parents to offspring. It is the study of heredity. What is heredity? Is the process in which traits. What do you mean by traits? Traits means characters. Characters are passed from parents to offspring. So you can see in this picture, this is a father and he has 3 children. So can you note some characteristic or a trait which is common between the father and the children? Yes, the nose. See, the father has a long nose and all the 3 children are having the same character. They have inherited this character, this trait of long nose. So this is heredity, in which the characters or the traits are passed from parents to offspring. Okay. Now, as I said, a character or trait is a distinguishing quality or characteristic, typically one belonging to a person. So you can see in this here that an individual having blue eye is a character, or a brown eye is a different trait. Okay, or a person can roll the tongue or another can't roll the tongue. So this is a character or a trait. Then what is genes? Genes control the characteristics of living organisms. They are in pair, right? So you can see this is a chromosome and this is a gene. So loci is the position of the gene on the chromosome. So this chromosome has multiple genes present which indicate the different phenotypes. For example, eye color gene located here on this chromosome gives the trait, the characteristic as a brown eye. Or another gene located for the hair on this part of the chromosome will give a distinguished character or the phenotype of the hairs, right? Now, what are allele? The genes of a corresponding pair are known as allele. You can also say that alleles are the alternative form of the same gene. So you can see in this picture that these are the 2 chromosomes, right? So one is from paternal side and the other is from maternal side. And here you can see the gene is located here from the paternal side for the hair color and from the maternal side for the hair color. So these are the alleles, right? These are a pair of allele. For every gene, you have 2 of the same allele or 2 different alleles. So remember, a chromosome with different genes present, and these are the alleles. One from parent, one from father, and one from mother. And these alleles can be same or they can be different. Very important to understand this homozygous and heterozygous. Homozygous means when both the alleles are same. For example, this is capital B allele and the capital B allele. So they are same. Small letter B and small letter B, they are same. So these are called as homozygous. What do you mean by heterozygous? When both the alleles are different. You can see here, one is having capital B and the other allele is a small b. Similarly, different. So these are called as heterozygous. Then, what is homologous chromosome? So you can see this is the paternal homologue and this is the maternal homologue. So a pair of homologous chromosome contains three of the features when to say that it is a homologous pair of chromosome. Number one, that both the chromosomes are of same length. Number two, the gene position is same. For example, the position of these two genes are at the same position, right? So this is having the homologous chromosome. These are the homologous chromosomes. And the centromere location. What is a centromere? This point is termed as centromere. This is the small arm and the long arm of chromosome. So this is a centromere which is separating the small arm and the long arm of the chromosome. So homologous chromosome is the same in length. The centromere are at the same position and the gene position is also the same. So we called it as homologous chromosomes. So in a pair of homologous chromosomes, one is inherited from male pattern and the other from the female. As I told you, what is the locus? It is the location of a particular gene. This is one gene. This is the other gene. They are indicating for the different phenotypes, right? And at each genetic locus, an individual has two alleles. This is one allele and this is the another allele. Okay. Now here to understand again what is homozygous and heterozygous. When both the alleles are same, we call it as homozygous. This is again the same, called as homozygous. And when they are different, see, one allele, the capital and the small letter, this is heterozygous. So I'll explain you what do you mean by dominant and recessive. But at this point, you just understand what is homozygous and what is heterozygous. So these are the three gene pairs at three different loci. What is a genotype? Genotype is the genetic makeup of the organism. The genes present in the organism is the genotype. And this genetic trait, when it is expressed phenotypically, when the character is expressed phenotypically, is called as the phenotype. The physical appearance which expresses the genetic trait is a phenotype. Very important to understand the genotype and the phenotype. Now coming to the genetic disorders can be broadly classified as disorders related to mutations in single gene. These disorders follow the classic Mendelian pattern of inheritance and also called as Mendelian disorders. There are certain single gene disorders even which they do not follow the classical Mendelian pattern. But here we are going to focus upon the Mendelian disorders which follow this inheritance. Okay. Then second classified are the chromosomal disorders. You know that we have normally we have the 46 pairs of chromosomes. 44 are the autosomes and 2 pairs of sex chromosomes. So if there is any alteration in the numbers of the chromosome like deletion or insertion etc., that is the numerical alterations, or there might be certain structural alterations. So they are termed as chromosomal disorders. And third are the complex multigenic disorders. In this, there is interaction of multiple genes with the environmental factors. Okay, now let's focus on the Mendelian pattern of inheritance. So here, this is Gregor Mendel. You must have heard his name, I think. He was a trained mathematician and natural scientist who discovered the basic rules of heredity. Now, out of curiosity, why children look like parents, he worked a lot on the peas plants. He experimented the peas plants because the peas were easier to work. He worked on multiple traits, the multiple characters of the peas. Like he worked on the seeds of the peas. He worked on the flowers, different flowers combinations. He worked on the shape of the seeds, then the pod color, the pod shape, and so on. So he kept on working for decades, and finally he became the pioneer in the field of genetics, where he showed that genes are passed from parents to offspring and get one allele from each parent, and so on. And so it is called as Mendelian inheritance. And the disorders following this pattern of inheritance are termed as Mendelian disorders. So under Mendelian disorders, we have autosomal dominant, autosomal recessive, and X-linked disorders. Before we go towards these disorders, let's study what do you mean by dominant and recessive. So, dominant means exerting a controlling influence on the expression of a trait, on the expression of a character. So, when the two alleles, here you can see when the two alleles are the same, right? So, it is homozygous. So, this homozygous, first you can take it clear that whenever we are writing a capital letter, it might be capital R, capital T, capital A, capital G, capital P, whatever. We take it as dominant gene. Okay? And that is when it is written in the small letter, it is a recessive gene. Make it clear. So here I consider that capital R is dominant and small r is recessive. So coming back to what is dominant, exerting a controlling influence on the expression of a trait means that could be homozygous. I told you what is homozygous means that both the alleles are same. So here both the alleles are dominant, means R and R capital, or they can be heterozygous, means capital R or small r. So if in case it is homozygous, both capital R or heterozygous, capital R and small r, these are the genotype. So phenotypically, the expression would be the same as the dominant character. And in recessive means the one that disappear temporarily. Now recessive might be RR heterozygous. So here heterozygous, or it can be homozygous. So remember, when recessive is heterozygous, the dominant character will be expressed. The recessive character will not express the trait. But a recessive allele will only produce a phenotype if there is no dominant allele present. The homozygous recessive. If both the alleles are small r, both the alleles are recessive, then it is going to express phenotypically. Otherwise, if it is heterozygous, the character present would be of dominant. If it is both dominant, obviously the dominant will be expressed. Okay, if both recessive, the recessive trait will be expressed. But if it is heterozygous, the dominant trait will be expressed phenotypically. It would be more clearer when we study the different exercises in upcoming slides. So, you should know what is Punnett square. So, to solve these exercises, this is a Punnett square. It is a square diagram. that is used to predict the genotype and the phenotype of the offspring. Now, very important to understand here. I'll give you one review problem to solve this Punnett square. First, let me explain you how to make this Punnett square. So make it clear that this Punnett square is made to understand or to predict what will be the genotype and what will be the phenotype of the offspring. Okay, now let us take an example of heterozygous yellow pea allele. Heterozygous, I hope now you understood what I mean. Heterozygous means both the alleles are different. One is dominant Y and the other is recessive small y. So here the dominant Y is giving the yellow color to the phenotype seed and the small y, the recessive, is going to give the green color. So when both the parents, parent 1 and the parent 2, both are heterozygous, means they both are having different alleles. One capital, one small, one capital, one small, means both of them are having one dominant allele and one recessive allele. Okay. Now you need to make a grid. So you can make a grid of like these 4 squares, and on the top of here of the grid, you write the alleles of one parent, that is this capital Y and the small y, and along the left side, write the allele of the other parent, capital Y and the small y. Okay. Now in the second step, what you do is, you combine the parent allele inside these boxes. So the upper one and the first from this parent you combine and it gives 2 capital Y. Then here we combine this and from this the other parent, this is giving the capital Y and the small y. And you combine the other allele of this parent and one allele of the first parent, and similarly these two both alleles are combined and you write down in these boxes. So these are the offsprings from these two parents. So this is giving the genotype ratio. What do you mean? Means that it is giving 1:2:1 ratio. It means that 1 is capital Y are 2 times. So Y1 and capital Y and small y, that is heterozygous, are 2, and 1 is homozygous recessive, year 1. So the ratio becomes 1:2:1. This is the genotype ratio. Okay. Now phenotype. I told you phenotype means that is expressed. These genotypes are expressed, or the characters are expressed phenotypically. So we know that, as I told you before, that capital Y, the dominant Y, indicates the yellow color of the seed, and the small y indicates the recessive green color of the seed. So when we see here that if the genotype here is dominant, both of them are dominant, so phenotypically it will express the phenotype character, the yellow seed. Then when it is heterozygous, I told you before, heterozygous capital Y means the dominant and the recessive. So the dominant character will be expressed. So the dominant is yellow. So these are the yellow seeds. But when both of them are recessive, then the recessive character will be expressed phenotypically. So you get the green seed. So you say that the phenotype ratio is 3:1. It means that 3 yellow peas and 1 green pea. Clear? Now again, to revise the Punnett square, this is the heterozygous dominant and recessive parent, and the other dominant and recessive parent are crossed, and we do the cross the same way as we did in the last slide, and these are the offsprings. So here you see that these are the dominant character. This is again would be the dominant because it is heterozygous, but the dominant is present. Again the heterozygous, the dominant trait will be present. This is homozygous recessive. So the recessive trait would be expressed. Homozygous dominant, homozygous recessive. These are the heterozygous. Heterozygous means both the alleles are different but expression will be of the dominant character. Please remember. I'll make you explain again the same here. When two heterozygous male and female are having brown eyes are crossed. What do I mean? That two heterozygous male and female. This is a male with a brown eye, but it is heterozygous. Means both the alleles are different, but it is expressing the brown eye. Means the expression pattern is brown, means dominant is brown. So I have colored it as brown. And the recessive, I took the example of blue eyes. You can take the example of green eyes, whatever, but the dominant is brown. Similarly, is crossed with a female, which is also heterozygous and having the brown eyes. So the dominant is brown eyes and the recessive is the blue eyes. Then I made the Punnett square and did the cross of these two heterozygous parents. What I found that 3 offsprings had brown eyes. So this is the homozygous dominant, the heterozygous and the heterozygous, but the dominant is having the brown. So 3 of the offsprings will have the brown eyes and the one individual with the genotype having both of the recessive character will have blue eyes. So only the person with this genotype, recessive, will have blue eyes. So please remember how it is inherited. The heterozygous parents are inheriting the 3 offsprings to be brown eyes and 1 offspring to be the blue eyes. So these heterozygous can be homozygous or 1 homozygous and 1 heterozygous. Whatever pattern can be followed. So the same revision here. Capital B is the allele for brown eyes, so dominant over small b allele. Small b allele is for blue eyes, so recessive to capital D, that is dominant. So possible genotypes will be homozygous, heterozygous or homozygous. When it will be homozygous, dominant will give the phenotype as brown eyes. When it's heterozygous, again the dominant is the brown eyes. When it is homozygous recessive, then it will give the blue eyes. Okay. Now we move forwards towards the autosomal dominant disorders. These are manifested in heterozygous states, remember. Okay. Now, So what are the characteristics of autosomal dominant? As I said that these are manifested in heterozygous state. What do you mean by heterozygous? When both the alleles are different. So this is one parent which is affected, but it is heterozygous. This is the mutated gene and this is the normal gene. And the other parent is having both the alleles are normal. So when they are crossed, here you can see this is heterozygous is crossed with the homozygous normal parent. This is the affected one. So when we cross, we see that 50% of the offsprings are having the disease. They are affected. See, these are affected. This is affected. 50% are affected. And the 50% are not affected. So here we say that the autosomal dominant is affected. So here at least one parent should be affected. Skip generations are not seen in this type of inheritance. Skip generations means the disease present is inherited in every generation. Then male females have equal chance of getting affected. See, the male to female ratio is same. Now the other characteristics you should know about is the autosomal dominant new mutation. It means that the parents are unaffected, the children are born, but a new mutation has occurred in one of the individual, in one of the children, and this mutation is during embryonic development. So this is having the autosomal dominant, means that this is in a heterozygous state. One allele is mutated and one other one is not mutated. So this is a new mutation occurred in one individual or one child of this unaffected parent, and this affected child can then inherit forward autosomal dominant in every generation. Okay, so with any autosomal dominant disorders, some patients do not have affected parents, and such patients opt to have the disorder as new mutation, and their siblings are neither affected. See, their siblings are not affected nor at increased risk for development of disease because the parents are normal, right? So this is the new mutation. The other features of autosomal dominant disorders are reduced penetrance. It means that some persons inherit the mutant gene, but that gene is not expressed phenotypically. That is termed as reduced penetrance. The gene is mutated in the individual, but it is not expressed phenotypically, right? This is reduced penetrance. Then second is the variable expressivity. It means that a trait is present associated with a mutant gene, right? A mutant gene is present, but that gene is expressed differently among the persons carrying the gene. For example, there are two individuals which are having mutant gene of neurofibromatosis, but one person will express neurofibromatosis as only brownish spots on the skin, but the other person having the mutant gene will express as tumors and skeletal abnormalities. So the same mutant gene in both the individuals, but the gene is expressing themselves in a different way in both the individuals. So this is termed as variable expressivity. Now the other character of autosomal dominant are they usually are presented in the delayed life. For example, in Huntington's disease. Then in autosomal dominant disorders, a 50% reduction in the normal gene product is associated with clinical signs and symptoms. Here you can give the example of like 50% loss of LDL receptors could lead to secondary elevation of cholesterol and then can lead to can then predispose for atherosclerosis in that individual. Now, the other characteristics of autosomal dominant disorder is dominant negative. What do you understand by that? For example, we take a collagen chain. So, as you know that the collagen chain is made up of three alpha chains. This is a trimer, yes? 1, 2, and 3 chains, alpha chains of collagen are twisted around it. So if one of the gene is mutated and the rest of the two are normal, what will happen? That the mutated gene reduces the assembly of the normal genes, right? Suppose this is a collagen chain which is mutated. It will reduce the assembly of these other normal chains, and this will produce the deficiency of collagen. This is called as dominant negative. Okay, this is called as dominant negative because it impairs the function of normal allele. And this example osteogenesis imperfecta, which is an example of dominant negative. So this is the list of autosomal dominant disorders which is given in your Robbins. So kindly go through them. You need to learn a few of them, at least 2 from this: Huntington's and the neurofibromatosis, polycystic kidney disease, then von Willebrand disease, hereditary spherocytosis, familial polyposis coli. Then these are very important to remember their names, which are associated with musculoskeletal disorders, and the familial hypercholesterolemia, very important. So these are the list of autosomal dominant disorders affecting the systems. So kindly learn them. Then we move towards autosomal recessive disorders. These make up the largest category of the Mendelian disorders, and they occur when both alleles at a given gene locus are mutated. Remember, in autosomal dominant, we said that the heterogeneous state, but here both the alleles are mutated and they are recessive. Okay, so here you can see that the characteristic says that offspring must inherit one copy of affected gene from each parent. This is the father and this is the mother. Both are the carriers. So the shaded one is unaffected and unshaded is affected, and when both of them are together indicates the carrier state. So this is the key. So this is a father where one allele is affected, the other is unaffected. So this is the carrier and the carrier mother. When they are crossed, what happened? They transmitted the disease only in 25% of the offsprings. So I said you that these are the heterozygous parent. We did the cross Punnett square, cross them, and what we find that this is a dominant character which is normal, unaffected son. Okay, then this is showing the heterozygous pattern. So this is the carrier children, but one of the child is having both of them as the recessive. So the both recessive is present, the affected daughter. So there is recurrence is 25%, 25% of 100 are affected. Male, female are equally affected in this. Here you see the skip generations. Yes, it means that suppose one generation is showing the disease, the other generation would skip the disease, but there would be the carriers present, and that carriers would carry the disease in the next coming pedigree or next coming generations. So this is called as skip generations, which are seen in autosomal recessive inheritance. Other features of autosomal recessive includes the expression of the defect tends to be more uniform than in autosomal dominant. Here you have complete penetrance. Then onset is frequently earlier as compared to autosomal dominant. Although new mutations for recessive disorders do occur, but they are rarely detected clinically, and many cases of autosomal recessive affects the enzymes. There are mutations of certain enzymes seen in autosomal recessive disorders. Okay, now this is again the table from your Robbins. Please learn the disorders. The most important, the cystic fibrosis, alpha-1 antitrypsin deficiency, Wilson disease, glycogen storage diseases. You should know their names. Then very important, sickle cell anemia and thalassemia are the autosomal recessive disorders. Yes. Then mutagenic muscular atrophies, Friedreich's ataxia, spinal muscular atrophy, and Ehlers-Danlos syndrome, some variants. Usually it is musculoskeletal, it is autosomal dominant, but some variants do show the autosomal recessive inheritance. Okay. Then we move towards the X-linked disorders. Now all sex-linked disorders are X-linked, remember, and almost all are recessive. There is no Y-linked inheritance. Please remember, there is no Y-linked inheritance because the males with mutations affecting the Y genes are infertile. So there is no inheritance, obviously. So a trait determined by alleles that are carried by the X chromosomes but are absent from Y chromosomes are called as X-linked disorders. X-linked disorders are mostly the recessive traits. Recessive traits controlled by X-linked genes are expressed in males. Again, very important to remember, in X-linked recessive, the males are the sufferers and the females are the carriers. We'll see in the next slide. And there are certain X-linked dominant conditions as well, but they are very rare. Just to remember that vitamin D resistant rickets is an X-linked dominant condition. Okay. Now here you can see I'm trying to explain you the X-linked traits. How we to understand why it is inherited in the females and not inherited in the males, and the females are the carriers. So non-gender related genes that are attached to the X chromosome but not found on Y chromosome. Women have XX, so they get two of these genes. Men have XY chromosome, so they only get one copy. So just focus over here. You can see this is the male and this is the carrier female. Carrier means one X is normal and the one X is affected. So when this normal male is mated with a carrier female, so we again draw the Punnett square, and what we see, the children here, we find that one male will not inherit any gene and will be asymptomatic. One female will not inherit any affected gene and will be asymptomatic. But one female will inherit the normal X and the other affected X would be inherited. See, inherited gene is present. But it will present with no symptoms. Why no symptoms? Because this normal gene is taking care of this mutated X here. It has masked this inherited mutant gene, mutant X over here. So the females will not present any symptom. But in male who inherit the normal Y but inherited this affected X will have symptoms because there is no other normal X to take care of this mutant X. Okay, so the females are the carriers. They do have the gene but they are asymptomatic, and the male have only one X which is affected and they are symptomatic. So the examples are the red-green color blindness, hemophilias, and muscular dystrophy. So if we meet this hemophilic father, suppose this is a hemophilic father which is affected with a normal female. Again, you can draw the Punnett square and see what progeny you get, the offsprings you find. You see that one male would be normal, the other male would be normal. So that both the males would be normal and both the females would be the carriers. Okay. So this is how the females one X takes care of the other mutant and they are the carriers, and the males are the sufferers in X-linked recessive disorder. Please remember. So again, to make you more clear and understand, you can say that, for example, this is a female having both X chromosomes, and you can say this is the B is the dominant and one small b is the recessive and is having the brown eyes. Yeah, heterozygous, but dominant is expressing. Let's say there's a recessive gene located here on this X chromosome, and that it is blocked. So this is the dominant. So this dominant will block the recessive and phenotype will be the brown eyes. But in males, you have one X and Y. Suppose this recessive gene is present here on the X, there is no corresponding gene to block this first gene. So the recessive gene is displayed even though there is only one. So the blue eyes will be expressed, right? So this was the sex-linked traits or the X-linked recessive disorder. These are the examples of X-linked recessive disorders. Please remember the names: the Duchenne's muscular dystrophy, hemophilia A and B, glucose 6-phosphate dehydrogenase deficiencies, then Wiskott-Ulrich syndrome, diabetes insipidus, Fragile X syndrome. You need to remember their names. You can learn from your Robbins. And that's all for this lecture. And I have given you this review problem. Try to solve them, make a Punnett square, and see if you how much did you understand. In pea plant, purple flowers. See, purple flowers are showing the dominant P characters, which over white flowers, which is showing the recessive. So remember, P here is the purple, P is capital, is dominant, and small p recessive are the white flowers. So you need to show the cross between two heterozygous plants. You say that one plant is capital P, small p, and the other plant is also capital P and small p. Make the Punnett square. Write on the upper side the capital P and the small p, on your left side again the capital P and the small p, and try to solve the Punnett square and see what ratio do you get for the genotypes and the phenotypes. Tell me the offsprings. So once you solve this Punnett square, write your answers, the ratio you get for the genotypes and the phenotypes in the comment box, and I'll try to discuss it in my next lecture, or if possible during your practical class.